think I've met, or definitely met Ian, but some of the other ones as well, maybe. So I first came to the lab in 2012, actually, as a volunteer. So yeah, it's 8 years ago now. So for all of those that are interested in volunteering, that's how I started. And then I've been back, or I was here for 3 years doing my PhD work, and then I've been back now for 2 years as president of the lab when Dr. Gruber retired. So yeah, we were looking forward to having everyone down here, but obviously this year is a bit crazy. So we're hoping that for next year it'll be able to go back to some semblance of normal and be able to see some of you down here in Bimini.
And my name is Chessie. I'm the outreach coordinator here at Bimini Shark Lab. So I first came to the lab as an intern as well in 2017. I interned for a couple of months before asking to come back because I loved my experience so much. So I came back the following year in 2018 before I was offered a staff and applied for a staff position in September 2018. So I've been here as the outreach coordinator for about 2 years. And my role is really discussing the research that occurs at the Shark Lab and breaking it down and disseminating our research to the public. So through talks, through public opportunities, through classes and Zoom presentations like this. So if you have any questions about kind of that side, it's a bit separate from the research track, but if anyone is interested in pursuing a career in education or marine conservation, I'm happy to answer questions about that at the end as well. So thank you to Ian for inviting us in to talk today. I do have a presentation that's just going to talk a little bit about the history of the Shark Lab. But if anyone has any questions to begin with, I will leave the chat bar open, so feel free to type in any questions throughout the presentation and I'll try and address them. And as I said, I'll have a Q&A session at the end. Peter, do you want to talk about the course now, or did you want to talk about it at the end?
I don't know, maybe I'll just say a word now and then I'll hang around. But so of course, I've been running a course, Shark Lab, every year except for last year for I think 12 years now. So that predates all of you. And except for Dean, who's not on the line. And it's a really great course. We go down for a week. We work with all these guys. It's about half or 2/3 sharks, but the rest is just all aspects of the ecosystem in the area and learning about the Bahamas. And the situation is, unfortunately, we had 16 students. We were all set to go last year, and then obviously it got canceled. Lining it up again for the coming year. I think the university will start advertising it next month. Obviously, the university's got to make some calls on policy for all their international courses. That's the thing you have to understand. But I'm proceeding and optimistic that we'll be able to have a go. And I don't know what else to say, but you'll hear all about the Shark Lab with Chessie. Did you want to say anything, Matt, at all?
No, yeah, I mean, so we're operating under the assumption that by, I guess it's like 10 months from now, that it's going to be in a better spot than it is now, like with the virus, and, you know, whether that's a vaccine or just like better testing that they can do. I know right now they're trying to come out with like tests you can do before even getting on the plane to the Bahamas. So hopefully it'll work out. And so we're operating under like the assumption that, you know, we'll be able to make it work for next year. But yeah, anything's possible, but we have to hope for the best.
Yeah, so I'm the same. We're trying same approach from this end. Yeah. And it should be advertised in about a month. And if you have any questions, just email me, Peter Sorensen, and I'll try and address them as best I can. Although I'm entangled in an incredibly complicated bureaucracy at this end, which puts a little bit more complicated than yours. So that's all I can say.
So we also have another special guest. Our vice president of the Shark Lab has also decided to come and say hi.
What? Little puppy. Oh, the lab knows the little puppies are a big part of it.
Matthew, what's this way? What's this way?
Say hi, bud.
Say hi to everyone. Yeah. So hopefully we will see some of you potentially here next year. if you do have any questions as well for us on the Shark Lab end, I will leave my email at the end of this presentation. So feel free to email myself or Matt regarding any questions you might have about interning or anything at the lab. But I'm going to get started and share my screen. Not do that. Perfect. Ooh. Perfect. Can everyone see my screen?
Yeah.
Yep.
Perfect. So thank you guys for inviting us to speak, as I said again. So I'm just going to talk to you a little bit about the Shark Lab, how we started, and a little bit about the current research going on at the lab. This is a lovely picture taken a couple of years ago of one of our tiger sharks that were caught on a long line. You can see Matt doing surgery there, but I always like to start with a picture of sharks to kind of give everyone a little insight into what it's like working with these animals. But to give you a little bit more information about the lab, we are a non-profit organization that was founded in 1990 by Dr. Samuel Gruber. He was originally researching lemon sharks out of the University of Miami. But due to a lot of development that happened along the Florida coastline, he also used to conduct and take part in research expeditions that used to go around the Bahamas. And while he was on one of these expeditions, he noticed that Bimini is a very diverse habitat, and a number of different shark species would cruise the shallow waters around the island, and realized that this would be a perfect base for his lemon shark research. And so this is around the late 80s and 1980s, and during that time he conducted some of the first telemetry studies right here in Bimini with sharks. And he set up the lab to be basically set up like a research vessel on land. So we're very different to some other places around the world. There are 20 people who live in the station at one time. We're actually currently going through a renovation, which is pretty exciting for any of you who have been to the lab previously or might be coming in the future. We're currently in the process of looking to build the lab, to rebuild the lab. And so at the moment, during COVID, we have been conducting a lot of construction work to ensure that the lab is here for the next 30 years to come. But really the main missions of the Shark Lab is firstly to understand the biology of sharks and rays and the role that they play in the marine ecosystem through research, field, and through laboratory work, and spanning multiple disciplines such as behavioral ecology, sensory biology, conservation, just to name a few. And the second mission of the lab is educating future scientists. So this is really where our internship program comes into play, our PhD students who are based at the Shark Lab, and through courses that come to the lab to learn about the work being conducted at the station. So really educating future scientists and providing opportunities for these students to learn field skills was a really crucial part of DOC's mission. And finally is to disseminate our research to the wider public through public tours, facilities talks, TV documentaries, social media, you name it. But this is really where kind of education plays a really vital role in spreading awareness about shark research and the research that's going on at the lab. But as I said, we are a non-profit organization. That means we aren't affiliated with any university. We're a Florida based registered, and we're also Bahamian registered non-profit. And that means we apply for research permits through the government, and we rely on external grants to fund our research. So one of our biggest funders is Save Our Seas Foundation, which is a worldwide organization that provides a lot of funding for different marine science research that's going on around the world. But the most interesting thing, and the thing that I think makes the Shark Lab such an amazing place to work, is the fact that we're 24/7. Because we're based on island, we don't have a normal 9 to 5 day-to-day life. We could be out conducting research at 2 a.m. We could be out conducting research at 10 p.m. It really having a lab based on the island means we can really look at the weather conditions and decide what research can be conducted looking at those conditions. Because if you think about potential other organizations or places that are landlocked, they'd have to pay in advance for a research boat, maybe sometimes up to a year in advance. You might get to 5 days before that research expedition and realize that the weather is just too bad for that boat to kind of depart and for the research to be conducted. And the beauty of us working from land is that we can pick and choose what activities happen purely based on weather and what that allows. But as I said, we do have a lot of students that are based at the lab. So how we run is we have some permanent staff base, and then we have rotating PhD students who are our principal investigators for about 3 to 4 years. And their role is to really fund, to ask research questions and move our research forward. So for the 3 to 4 years that they're based at the station, we focus on their research projects, and then when they leave, we'll have another rolling 3 PhD students who will come in to take over from them. But over the years, we've had over 20 PhDs be conducted at the lab, 37 masters and 13 undergraduate theses, and numerous collaborations with professors and scientists from around the world, not just in sharks. We used to be called the Shark Lab, but we've renamed to Biological Field Station, as we have visiting scientists who might be studying a number of different species found on the island. But as I said, we've published over 100 scientific papers to high impact journals, offered over 1,000 internships, and tagged over 8,000 sharks during our 30 years running. But just to talk a little bit about kind of why Bimini is such a great place for Shark Lab to be based, it really geographically, it becomes really clear when you see if you look at this satellite image right now, you can see we're only 50 miles from the coast of Florida, and we're separated by this really deep blue water, which is the Gulf Stream. That brings fast moving water right past Bimini. You can see we're very, very close to this dark blue line here, which is the drop off. So that's where the water drops to kind of 300, 400 meters from the shallow waters around the island. This is the area where my mouse is right now is the Great Bahama Bank, and it's miles and miles of very seagrass rich, very, very shallow warm water. And in comparison, on this west side of the island, you've got the Gulf Stream, not very, very close to the island. So every time the tide changes, we really get nutrient-rich water deposited right at the island, and it really makes Bimini a biodiversity hotspot. And a number of different species are found in these different habitats around the island, from the seagrass flats right to the sandy bottom on the west side of the island. So just to show you a little bit of what that looks like, this is a video taken from a drone from one of our mangrove spots on the east side of the island on North Bimini. And you can see, these are sprawling kind of red mangroves, and you can see a number of different fish will use this area and cruise these shallow waters, using it for protection and food. And you can most importantly see these juvenile lemon sharks swimming throughout these mangroves. And in comparison, this is to the south of the island. This is a baited remote underwater video system, or BRAV it's known for short. So it's basically a GoPro linked to a bait crate, and it was set for a certain period of time, and it's basically a way we can use to measure abundance in an area. So it's a kind of a way that you don't have to have anyone in the water to kind of count the number of fish. It's a really effective way of doing that. And you can see a number of these different fish species found on the reef system here. So you've got grouper to armico jacks, to queen triggerfish, to a number of different schoolmasters and snappers. So it's a really, really beautiful habitat with a number of different species. But really, kind of why the Bahamas is known really as a shark hotspot around the world. A lot of people pay a lot of money to come to the Bahamas to swim the sharks and to swim in beautiful waters and visit the nice beaches. But really, Bimini and the Bahamas in general has a number of different species that most people probably aren't aware of. And a lot of these are some of the deep sea species that you can find in some of the deeper areas. So for us on the west side, as I said, there's that drop off into the Gulf Stream. So occasionally we conduct deep line surveys there, and we can catch stuff like Squalus cubensis, so you can catch stuff like Cuban dogfish, dusky smoothhounds, even sometimes six gills around that area. And then if you go into places like tongue of the ocean as well, you can get some of those more migratory species as well. You can find stuff like silky sharks, Cat Island, you can find species such as oceanic whitetips. And actually in the Bahamas, we have over 40 different species of sharks, representing 7 out of those 8 orders. So it's pretty incredible the number of different species of shark that we have around the island. And just to touch upon why that is important, not only for us, obviously we're conducting research, but for the Bahamas in general, we're very lucky that we have a very supportive community here in Bimini. But on the larger scale, the Bahamas is known as the shark diving capital of the world, and diving in itself provides a lot of money to the local people and the Bahamas. And actually a study conducted in collaboration with the Cape Eleuthera Institute, which is located in Eleuthera, a couple of years ago in 2017, actually estimated that the shark diving industry in the Bahamas is worth $113.8 million every year to the Bahamian economy. Now for an island, and especially some of the outer islands like Bimini, which aren't particularly wealthy, that's an incredible amount of money generated just from this diving industry. So it's definitely, sharks are very important. In the late 1990s, the Bahamas actually banned commercial longlining here in the Bahamas. And that meant, you know, there was no longer these long lines to catch tuna and some of the big game fish kind of around the island, and instead recognized the value of that one family visiting an island such as Bimini to go out and sport fish for that one weekend, and then they go home and tell their friends that they had such a great weekend sport fishing. The Bahamas recognized that they generated families than they did necessarily from those commercial fishing operations, and therefore banned the longlining in the late 1990s. And then later in 2000 and 2011, the Bahamas was made a shark sanctuary. So it meant it's illegal to kill sharks around in Bahamas. It's legal to catch them. And which really has kind of raised the profile of sharks in the Bahamas. And for us as researchers, it's meant that we have access to a number of different shark species that cruise the waters around the island, and we don't have to worry about commercial longlining operations in the area. But why is that important, and why do I bring that up? Well, Doc has always been very vocal about conservation matters. He's been in Bimini since the 1980s, conducting one of the first telemetry studies, as I said. But really, when he built a lab in 1990, his vision was to create a world-leading research station that was multidisciplinary and studying a number of these different species. And it's incredible that we have the diversity that we do here on the island. So actually, just to talk about these images right here, I think this was taken in the late 1990s, and this is in comparison, this is a picture taken from last year with our principal investigator Clemency White, and she's conducting a behavioral trial on some juvenile lemon sharks that are in this pen right here. As you can see, we use pen mesh sometimes to hold juvenile lemon sharks in semi-captive environments. That means that they're not closed off from the ocean, the water, and they're subject to tides in these pens, and small fish can enter and leave. But it means we can actually use these pens to conduct behavior trials and answer different scientific questions and keep these sharks in a captive environment and release them into the wild and explore whether or not behaviors change, for example, in a wild environment as well. So Bimini is we're very, very lucky to be based on an island that has shallow waters and access to a population like these lemon sharks. But the lemon shark is really what Dr. Gruber's early work was on. He started some of the first early growth studies on lemon sharks. This is a picture of them injecting a dye into the vertebrae of this lemon shark. It's called, I believe it's tetracycline. Apologies if I've got that wrong. Basically, the way we age sharks is, unfortunately, you do at the moment have to sacrifice that animal. But with the early growth studies, what you're able to do is inject a dye into an animal, and that dye into the vertebrae column. And sharks, a bit like trees, lay deposits down within their vertebral column. And if you inject the dye, it leaves a deposit within that vertebral column. And if you go back and recapture that shark certain time frame later, you're able to look at where that mark was left in the vertebral column and count how many of those deposits were naturally laid down by that shark. And therefore, you can kind of estimate how old that shark is if you know how long it took them to lay down those deposits. You can pretty much count it like rings on a tree. He also did some of the early work with a respirometer, working out how much the metabolic activity of lemon sharks, how much oxygen they were used in day versus night. That's actually a very interesting study that we've actually recently redone. We had a collaborating scientist, Evan Bryans from University of Murdoch. Doc conducted that study in I think it was 1980s, or 1980s, when he was at the University of Miami with sharks between 1 and 2 years. And Evan has recently released a paper about, for his PhD defense, using an older age category. So it's really exciting that we can see some of Doc's early work moving forward 30 years later. And again, some of the early learning experiments, he was very key for conducting some of those, and that really has pushed our science on. So everything we really know about the lemon shark pretty much came from Dr. Samuel Gruber and the work conducted prior to the Shark Lab and at the Shark Lab with his PhD students. So you might be asking, how do we conduct research on sharks? It's obviously not an easy animal to work with. Unlike terrestrial animals, that it's a lot easier to keep in captivity. That's not something that's feasible a lot of the times with certain shark species. It might be for species like the lemon shark that we've got here that do very well in captivity for a short period of time. But we have species that are found around the island that are critically endangered, such as the great hammerhead shark. That's obviously a fork that can get up to 4 meters or even larger in some places. That's obviously not feasible to keep in captivity. So how do we conduct research on sharks, and what is the Shark Lab doing at the moment? So firstly, standard with kind of most scientific protocols, it's really about the questions you're asking, whether or not you understand your study species, how you can answer the question, what method you're going to use. And then when it comes to sharks, also there are a number of other things you need to consider, kind of what capture method is best suited, what data you need, what can that data tell us, and what is the implication of this work? Is it important? Primarily being your one of your important questions to ask. And again, then you go forward with data analysis and your output, and what's the future directions from a study. So at the moment, as I said, we have 3 PhD students who are studying a variety of different things. But one of the things that all of our scientists at the lab have to do is conduct a workup. So this is data collection on an animal. So I've got a short video here for you, which will show you a little bit about how some of this data is collected at the lab. So this is Vital Heim, one of our PhD students. He is studying the great hammerheads here in Bimini. And this clipping is just showing him working with some of the great hammerheads, IDing them for a study. He's looking at the impacts of provisioning on the great hammerhead. So that involves a lot of scuba diving. In comparison, some of our juvenile species, such as the lemon shark and the nurse shark and stingrays, we can keep in semi-captive pens that I spoke about earlier, and conduct workups through blue tubs on the beach or on a boat before releasing that animal or keeping it for a short period of time in those semi-captive pens. And we can take samples such as blood, DNA, isotope samples, which is basically can be used for diet analysis. And we can assess the whether or not that individual is pregnant. So this is an ultrasound that was given to us by EI Medical. That's looking into whether or not the ray is pregnant, and we've just got a picture of a bull shark here. And we can use that and then there over the side of the boat to find out whether or not an animal is pregnant. So really, really useful for us in the field to understand a little bit more about the animals we're working with. And as I said, we take blood from our animals. In some cases, we'll even put tags on them, such as satellite tags or acoustic tags. We'll take length measurements to kind of assess how big the shark is, and girth measurements to assess body condition of that animal. But really, one of the key things for us is we have a variety of species, and each time you're working with a species of shark, you have potentially might have to use a different capture method to successfully catch that animal and to work with that animal. Just going to pause that. So as I said, we conduct research on sharks, and we take a number of different samples. So for us, and taking some of the important samples we can take is a fin clip. So that's a small little clipping from trailing edge of a dorsal fin of a shark, or of the pelvic fin of a stingray, for example, and we can use that sample to tell us about the DNA. So one of the important studies that came out of the Shark Lab was actually a study done on the lemon sharks, and that's because of a 30-year data set we've collected over since 1990. And that data set, basically by taking DNA from every juvenile lemon shark that was born in Bimini, we've worked out that actually a lot of them are related, or we can basically create these family trees. So how that works is every time we catch a juvenile lemon shark, we'll put a tag in it, and we'll take a sample, and we often recapture these individuals. And from what we noticed was the juvenile lemon sharks that we might have in the 90s, we might have caught them 12, 13 years later as fully mature adults on our long lines, instead of being in the mangroves where we find them as juveniles. And what we've realized is we might also catch some juvenile lemon sharks in those mangroves that are related to a large female that we caught that's come back. So we can really basically match up the different individuals we find around the island and create this whole family tree of the lemon sharks. So think of it like ancestry.com for sharks. It's pretty cool. And that's purely because we've got this 30-year data set and basically found out that lemon sharks conduct something called natal phylopatry. So pretty much similar to turtles, they come back to where they were born to give birth to their own young. So lemon sharks are born in the mangroves around Bimini in these different nursery sites. They grow up in these nurseries, leave when they're of a large enough size, and then the females will return 12 to 13 to 14 years later when they're fully mature adults to drop their own pups in the same nursery site that they were born in. So it's a really useful sample we take. We can also use that fin clip to, as I said earlier, to find out what these sharks have been eating. We can also use this small muscle sample to find out the same thing. So what these sharks have been eating recently. We can also use blood to tell that as well. And we can also use blood to tell us assess reproductive hormones, stress. It's a very useful sample that can tell us a number of different things. And the same way we can do an ultrasound, as previously mentioned, and we can put tags out on these animals to find out where they go. So what does some of that look like? As I said, we put tags out. These are I've got 3 pictures here of some of the sample tags that we use on some of the animals, but it really depends on what research question you're asking about which of these tags you use. So the first is this first picture here is a Casey tag, or a no external Noah's fishery tag, or M tag. And all it is an external marker. So this is a small dart that kind of sits in the muscle layer and by the dorsal fin. It's attached to this monofilament here, and a small capsule at the end that has a unique number. And they come with a card that goes that gets filled out with the information when you catch that animal about its total length, where you caught it, and that gets sent off to NOAA. And what these are used for is purely external identification. So if they get caught accidentally in a commercial fishing operation, the person who caught that animal could open up this capsule, find out where they can send the information about that shark off to, and we can get notification that our animal was caught elsewhere around the world. So, but most of the time it's purely for external identification purposes, and it also tells people that this is a research animal. In comparison, the 2 tags I've got at the end here, this is what we call an acoustic tag. And these, we have different variety of different types of these tags. Some of them have different battery life compared to others. Some of them can detect pressure, and these get surgically implanted inside an animal. And how they work is they're passive acoustic tags. So they don't, they're not tags that you need to follow with a hydrophone, for example. They're actually paired with underwater listening stations or receivers that are placed on the seabed floor at different depths all around the island. So we actually have currently 68 deployed around the island. And whenever an animal that's got one of these tags in swims within 200 to 400 meters, depending on how deep that area is and upstream is like, that receiver will record that individual tag. So each of these tags have a unique ID number and serial number. So when we go to download our receivers every 6 months, and we basically clean the data, and we can find out which individuals are on each receiver. Now we record what's the habitat type, where each receiver is deployed. We can also match up basically what the local space use is of that animal. So if we know it's a tiger shark that has one of these, and we find it on the east side of the island, we can see whether or not it was recorded on any of the other receivers in that area, and basically note the local space use of that animal. Now, the cool thing about these tags is it's not just us that use them. It's a number of different research organizations around the world that use the same tags made by the same companies. They're made by a company called Vemco. And it basically means that any animal that has one of these tags in them can be detected by our receivers. So for example, if a scientist or a research institution in the Carolinas put one of these into a tuna, for example, and that tuna swam past one of our receivers in Bimini, potentially our receiver could pick up that tag. And when we go to download our data and clean it, we can send off information into a larger collaborative network that's around the world, or in this area. So the FACT network, for example, is the one that we use most. We can share information that their research animal was found in Bimini, and vice versa if one of our animals was detected on another institution's receiver array. Now, these are really, really useful for a lot of the species we have around the island, such as tigers. They're put in great hammerheads, the lemon sharks around here, the bull sharks. Some of the southern stingrays have these tags in as well. But for some of the species and some of the work that's going on at the lab, we're actually trying to find out a little bit more information about where these sharks go. And a lot of the sharks, such as the tiger sharks and the great hammerheads, might utilize space in the middle of the Atlantic. The limiting factor for these acoustic tags that we use with sharks here is, unfortunately, you need to rely on receivers being placed on the seabed floor wherever those sharks are swimming. Now, that's great along the coastline of the east coast of the US, for example, but if we have a shark that was tagged in Bimini and goes straight into the middle of the Atlantic, there's no way we're going to be able to put receivers on the seabed floor that are going to detect that tag. So an alternative method we use are the satellite tags. So this is where these satellite tags come in. So we use something called spot tags. There is another type called PSAT. So but we use a fin mounted tag instead. And how these work is you can think of them like strapping an iPhone to the dorsal fin of a shark. And for our highly migratory species, such as the tiger shark and the great hammerhead, they can tell us where these sharks are and pinpoint the location of these animals when they're in the middle of the Atlantic, as long as their fin is out of the water. So for species like great hammerheads and tiger sharks, for example, they might be, for a tiger shark, there might be a whale carcass in the middle of the Atlantic. And while it's, if a tiger shark comes across that, a lot of the time it will eat that whale carcass. And while it's feeding on that surface, its fin will be out of the water. And there's a little sensor at the top of this satellite tag that when it goes dry, it then tries to locate a satellite, and basically we get an email from our shark. So just to show an example of this, I hope everyone can see my screen still.
Yep.
Yeah?
Yeah, we can still see it.
Okay, amazing. Apologies, I'm just going to zoom in.
Oh, actually, maybe not anymore. We can see the presentation still, but not the website.
Okay,
I
apologies.
think you might have to hit share screen again, and then it will like.
I'll try again.
Like switch to the website. Yeah, now we can see the map.
So this is a tiger shark that was tagged for President Matt's tiger shark work last year. So it's tagged in Bimini, and all of these points, these blue points, are where the shark, we got a signal from that shark. There's obviously a slight location error. The shark obviously didn't cross land right here. But between where the shark last left the surface of the water and the next one, it's drawn a straight line. So you can see that it spent a lot of time around Florida, kind of be a little bit further up from the Bahamas before kind of swimming down, and then we believe it went right into the middle of the Atlantic, really, and all the way over to the west coast of Africa. So that's a pretty incredible track that you can get, and information you can get about the movement of some of these highly migratory shark species. I think this one, the tag finished the first location, so it was tagged in 16th of June 2018. And its last location, so we got a full year, just over a year worth of data from this tagged tiger shark, which is pretty incredible. I'm just going to stop share and go to... So you might be perfect. So you might be asking kind of why I'm kind of showing you this. I think it's important to highlight how difficult it can be to work with some of these shark species. So this is us, a recent video of us putting a satellite tag on one of our tiger sharks. So we work up all of our large sharks over the side of the boat. So this was one caught actually last week that currently has an active tag, satellite tag deployed on its fin. She was a 290 meter female, so she's not quite sexually mature. We ultrasounded her to see whether or not she was. Normally for our tiger sharks, kind of 3.3 over 3 meters, you would think that she might be reaching maturity, but she's definitely immature at this stage, but we think she's on the cusp of maturity. She had a lot of scars down her sides that was kind of indicative that could potentially be mating scars, but as I said, we think she might be a little bit immature. But Matt wanted to really find out where she was going, so we placed this fin satellite tag to her dorsal fin, and we basically all do it over the side of the boat before releasing the animal. So this was a 15-minute workup. So we collected all the samples earlier. Ultrasound satellite tag was put on. We also put one of those acoustic tags inside her, so surgery to put one of those 10-year tags inside her before releasing her. And we've got quite a few coordinates and track from her so far for the past 2 weeks. Just put that on the side. But really what the current research projects ongoing at the station and the ones that our volunteers are integral for helping us with. So when you come as an intern to the station, your role is really to be the research assistant at the station. And when we have university courses at the lab, you get to take part in the research ongoing at the station. But the first one is the fishery conservation and ecological role of great hammerheads. So this is work conducted by Vital. He's wanting to understand more about where great hammerheads and scalloped hammerhead sharks go, recently reclassified as critically endangered. While they're in Bimini for the great hammerheads for a very short period of time, December to April is considered winter season for them. While they're around the island and feeding, for example, there's this dive tourism, there's a diving operation here in Bimini that feeds the great hammerheads while they're here. While they're using the waters around the Bahamas, they're definitely protected from some of those commercial fishing operations. We know that great hammerheads tend to go north once they leave Bimini when the water starts to get a little bit warmer. And what Vital really wants to find out is whether or not there's a crossover between areas that commercial fishing operations are using and these great hammerhead sharks are using, and whether or not there's potential to discuss a time area closure. So you could ask fishermen, this commercial fishing operations, to use potentially different gear, or to use a different capture long lining method and different equipment to select against hammerhead sharks. So that's really what his PhD work is looking at around here at the moment. He is also conducting an impact of provisioning study at the lab, so looking at the different individuals that use the shallow water around the island. We're actually able to ID them based on markings, scars, pigmentation patches, notches in the dorsal fin, and you can tell, he can tell pretty much 58 of the different sharks that are individually recognized here in Bimini apart. And he can work out then how much food they're taking on these commercial on these dive operators, with Neil Watson, one of the scuba diving operators that are feeding the sharks, and work out what how much of food these individuals are eating, and whether or not we're changing the behavior of these sharks that are coming in. You can imagine provisioning is a very important topic that concerns divers and scientists alike. We always want to know what impact we're having on a species, especially a species that is critically endangered. So really interesting work that's being conducted right now. Our second major project is Matt finishing off Matt, our president's PhD, which is looking at the ecological role of tiger sharks in Bimini. So in the wintertime, we tend to find large mature females cruising the shallow waters around the island. And then during the summer months, we tend to find juveniles, so 70 centimeter plus tiger sharks, which are tiny. It's very, very cute. But why do we find the larger females during the winter to spring months, and we tend to find juveniles or immature individuals in the summer months? That's definitely something we want to work out. Why is there that partition? And one of the suggestions is that some tiger sharks will use the Great Bahama Bank and the shallow seagrass warm waters to pup during the spring months, and that's why we tend to catch more juveniles during summer. So he's looking at that's why he's tagging so large sexually mature females to find out where they're go to see if he can work out the space use and where these animals are going. And our final project is by PhD student Clemency White, who's looking to anthropogenic impacts on sensory capabilities of the lemon shark. So we've moved on conducted quite a lot of research on the lemon shark over the past 30 years. But what Clemency wants to find out is what impact are we having, and what impact is boat noise having on the sensory capabilities of the lemon shark. So we recently just finished another set of behavioral trials for her a couple of weeks ago, where she was assessing the rate of movement, whether or not the sharks were stressed when there was the application of a boat noise in comparison to when there was no noise or no external stimuli. What was the difference in the rate of movement and the areas that they use? So she's doing a study in semi-captive environments, where she'll assess that using those semi-captive pens that we spoke about, and using an engine, and looking about where those sharks, what space those sharks will use when there is the presence of a boat engine noise and when there isn't. And she'll also be putting tags out on these juvenile lemon sharks, and observing that behavior in the wild. So she'll put acoustic tags that are active acoustic tags, so you basically have to follow them with a hydrophone to see the space use in these two different areas. One is a pristine environment that doesn't have much development going on, and the other is in our North Bimini, where there has been a lot of development and mangrove removal. So these are the current research projects ongoing at the station at the moment. But as I said before, we have quite a lot of other research projects that are involved in kind of over the 30 years we've been established. So our pit survey, which is the juvenile lemon shark census that happens every year. We obviously have the hammerhead work, the impact of provisioning, which is was started as a master's project in 2016-2017 by Vital, but is being continued each year subsequently, so we can find out whether or not the behavior of these great hammerheads is changing year on year. And then another really important thing for us at the moment is apologies, I'm just skipping through these, is the effect of habitat loss around Bimini. For those of you who haven't visited Bimini, Bimini used to have mangroves all along the north of the island, but over the years there's been a lot of development that's happening on the island. So this is a picture from 1980, where you can see there's mangroves all along the west side of the island, so all the way along this area here. And in 2003, a lot of development happened to build, to remove the mangroves and to build a brand new hotel. In 2005, that was kind of continued upwards along the west side of the island. So this point here is pretty much this point on this second image. So you can see there's been a lot of mangrove removal that's happened in Bimini, and the hotel has been there for a number of years. They're even conducting more work right now that's removing all the mangroves right up to the very tip of the island right here. But sadly, and I've got a video to show you, this is for any of you who have visited the lab, and Peter, you might see this. This is something that happened during quarantine. This is South Bimini. This is a nursery site for juvenile lemon sharks, and this is a special site called Duck Pond, where one of our board of directors conducted his PhD assessing the space use of juvenile lemon sharks around here. They used to use this area a lot, and it has changed over the years. And this video just shows you what's happened over the past couple of months this year. Yeah. So this is prior to the removal. So this is what it looked like in January 2020. So the beginning of the year, there was mangroves all along here. And this is what we call Duck Pond. It was separated, and this is what it looks like today. And this is right by the Shark Lab. So this is literally, if you stand on our beach, you can see this from our beach. And this is what's happened in the past couple of months. And you can see the extensive removal of that mangrove habitat, the closure of that duck pond, which is a refuge on south for those juvenile lemon sharks from predators that might come close during the high tide. And you can see kind of all of the silt kind of going into the water around the island. So this is our channel. This is where you enter on the boats to go into the channel from where the Shark Lab is. And this whole area was used by a number of different species, snapper, southern stingrays. We used to catch juvenile lemon sharks across here. So it's really sad to see that development is still kind of going on in Bimini to such an extent that it's kind of out of proportion with the size of the island. Bimini is a very, very small island. So yeah, I thought that might shock you, Peter.
Yeah, it does. I'm really horrified.
Yeah, it's really sad to see. And for us, mangroves are also protection from hurricanes. We're very lucky to be spared this year from any of the major ones that came through. Hopefully we've still got a couple more weeks of the season left. But it's really important for us that what work we're doing can be used to inform government, inform policy, and to try and stop removal of important habitats. And it's not just for sharks and fish species around here. There are a lot of fishermen that rely on the mangroves economically for lobster, snapper, crabs, conch. They're incredibly important, diverse ecosystems that are important for a number of livelihoods. So it's really important that we monitor changes in these environments so we can best inform. But yeah, so it's really sad to see. And Ian, I know you were at the lab last year, but it looks very different now on South Bimini. And there is talk that this is potentially going to be turned into an eco resort. Which I find quite funny that it's going to be labelled as an eco resort when they removed the natural habitat to build it. So that kind of brings me to the end of my quick kind of overview of what's going on at the Shark Lab. I'm more than happy to answer people's questions. I do have some videos that I scrolled past. If anyone would like to watch them as well, I'm more than happy to share them. But if anyone has any questions, either feel free to unmute or ask your questions. I also have invited Ellie Richardson, who is our station manager.
Hello, not sure if you can see me.
I'm going to stop sharing my screen, apologies, and come back on here, who will be more than happy to answer any questions you might have about interning or life at the Shark Lab in general.
Yeah, hello everyone, thanks for tuning in, nice to meet you all. Yeah, mainly if you have any questions about volunteering, I can answer those for you. I am the station manager here at the lab, so one of my main responsibilities is hiring and then overseeing the experience of our volunteer interns. Just to kind of give you an overview of that, I know Chessie's introduced you to the lab, but maybe hasn't touched too much on the volunteer program itself. We have usually around 10 volunteers stay with us throughout the year. There are specific dates that people leave and start the internship, which are not that flexible, so do check out our website for those specific dates. For the most part, they fall into winter, spring, summer, fall, so kind of those 4 seasons there. And then you stay for 2 months. Most of the internships are a minimum of 2 months, but you can stay for up to sort of 6 to 9 months if you really wanted to. But it's a really good opportunity if you're interested in science, sharks in any way, or just kind of looking to network. Maybe you don't know what the next step is in your career. You might just be starting university, or you might be towards the end of your course. It's kind of suitable for a whole range of people to come here. And we do try and hire people from kind of a vast array of experience and backgrounds as well. So we kind of have a nice group dynamic too. So if you don't feel you have a lot of experience on your point, don't let that put you off applying. With that being said, it is quite competitive, but competitive in the sense that we want to see you putting a lot of effort into your application. So if you are interested, make sure you're putting details in your CV. We do ask questions about teamwork and things like that, but it doesn't have to be specifically teamwork experience you have in the science world. You know, it could just be in another job that you've done as well. So include as much information in there as possible. We do have the opportunity for you to add letters of recommendation. Maybe Peter will kindly write you one. Cover letters, your CV. You know, there's quite a few things you can do to bolster up your application as well. And I know some of you have been to the Shark Lab as well, Ian. So you kind of so maybe you can kind of tell people a little bit more about what to expect. Although I'm not sure if Chessie mentioned, but we have been doing some renovations this year. So even since then, the Shark Lab has kind of been polished up a little bit. But for the most part, it's dormitory style living. So that's something to consider as well. If you think you can kind of thrive in that environment, bounce off the same people that you're working with, it can be quite intense if you're not used to that. Although if you're at university, you probably have experienced some dormitory style living already. So that'll set you up well. But yeah, it's a really inclusive program. We're very much a team here at the lab. The staff and volunteers are together all day, every day, doing science, but also the flip side of things, you know, cleaning and the maintenance of the lab that kind of goes part and parcel with working on a remote island with limited amenities. But it's a really fun experience. People from all walks of life have come through and kind of gone on to do various things, you know, gone to do their PhDs, or maybe they're interested in outreach and education, or perhaps we have quite a few people that come through and realize they like veterinary science, for example. So people kind of go off to do all sorts of things once they leave here as well. But the main thing we can offer you is really great practical field experience. So we are a little bit limited in the sense that we don't have a working laboratory on site. So all of our samples get processed and analysed out with the station. We send them back to the US or to Canada. So if you're looking for kind of lab technique experience, we can't offer you that. But if you really want to get out there and utilise practical skills, get out in the field, out in the boats, have some hands-on experience, and that's pretty much what your entire internship would be here. So in which case it would hopefully be very, very suited to you. We do hire a little bit in advance. So right now we are fully booked until April for interns. The April deadline is actually coming up in a couple of days. So if that's a period you're interested in, start thinking about looking on our website. I think it's the 15th, so you've not got much longer. But the summer is well open. I think the deadline for that is January. So usually applications have to be 6 months in advance. So you do need to know your schedule a little bit. Yeah, that's pretty much an overview of the volunteer program. If you do have any specific questions about that, more than happy to answer as well. But yeah, and if you've got any experience working in a remote environment, or as I said, traveling or dormitory style, that kind of all contributes to you being a suitable volunteer as well. So I just took over.
Real quick, I just wanted to say thank you so much. It was a really interesting presentation. I have another class starting soon, so I'm gonna have to leave, but I just wanted to say thanks, and it was great to meet you guys before I have to head out. So
No problem. Thank you so much for tuning in.
Nice to meet you.
Does anyone have any questions about the presentation or about volunteering or about anything you saw today?
Yeah, I have a question. So you guys have so many projects going on at one time. Do you all kind of pitch in with each other's projects? Or are you focused on your own project? How does that normally work?
So what happens is we normally have a staff meeting in the morning where we assess what the research priorities are for that week. So our scientists will say, I need to get a certain number of tags out, or try to. I would like to go and bait for hammerheads, for example. I want to try a long line. We just assess what the priority is for that week. And then as the staff team, we'll try and ensure that we make all of those priorities happen as long as the weather allows us for that week. So basically our interns will swap around. So one day they might be working with Clemency doing lemon shark work, the next day they might be with Vital, the day after they might be helping doing maintenance and scientific maintenance and back at the lab, and the day after they might be helping with a long line. So it's really versatile work environment, and everyone pretty much helps everyone. That's one of the cool things about working. You might be, really interested in kind of sensory biology, but one day you might be working with Clemency for a couple of days in that week, but you'll also get experience out in the field working with great hammerheads and tiger sharks and doing some of our movement ecology stuff. So it really just depends on what research priorities are there for that week or that month. But it's quite exciting. So you never know what you're going to be doing each and every day.
It's probably worth mentioning as well, we do have project student positions too, which usually only come up a couple of times a year, but the role of a project student is to assist Clemency and Vital more closely than the other interns. So as a regular intern, you kind of get, as Chessie said, a very all-rounded view of the science here. As a project student, you still get an all-rounded view and you kind of get to dip your toes in everything, but you do have more of a focus assisting with Clemency or Vital's project, and alongside their research you kind of have your own side project as well. So if you are interested in having something with more of a focus and kind of bouncing off of our principal investigators, that's something to look out for.
Yeah.
Yeah.
And normally those project students will kind of get a paper or will like defend a master's or something, so there'll be some sort of scientific output that will come from the work that you're helping with, and normally if you've helped with Clemency and Vital as well, it's like acknowledgement on papers and stuff that come in the future. So it's really great if you're looking to stay in academia and move forward with a research career.
Yes, Peter, I'd just like to say, I think close to a dozen people from Minnesota have now interned or been managers at Shark Lab
Quite.
over a long time. It's quite a few, and it's always gone really well. It's a great opportunity. And if you're still not quite sure, I just wanted to say, if you can join my course next year, we always get a pretty good flavor for it as well. And a lot of people use that as springboard to become an intern or even go on to grad school.
Yeah.
A number of people have done that.
And I would, following on from that, Peter, also for us and for Ellie, when she's assessing interns as well, like if you've come on one of the courses as well, and you normally talk to her and make it known that you're interested in volunteering as well, it means that when your application comes up, she's already got a face to a name as well. So which is always a really good thing as well. So it's a really great opportunity, the course as well, if you're interested in doing that in the future, to really kind of come here and like let us know what you're interested in and show your enthusiasm and your excitement. So that is also a really good way. We get quite a few people who come on courses who do go on to be interns with us as well.
How did you guys get working at the Shark Lab? Like how did you do your studies? How did you find the Shark Lab? How did you get started there?
So I knew about the lab from quite a young age. My mum grew up going scuba diving, so she used to scuba dive for a number of years, and every time we used to go on holidays snorkeling, we always used to watch like nature documentaries, and I think we came across the lab. I can't remember, I must have been 9 or 10, and I was like, that's so cool. And I can't remember exactly, it was on the BBC, but it was a very old one of doc, so I always knew about it. And then I thought I wanted to go into veterinary science, so I spent most of my kind of teenage years and pre-university years interning at different animal involvements of farms, veterinary practices. And then when I kind of got to age when I was thinking about university and college, my kind of interest in marine education and conservation had never gone away, even though I was trying to pursue a career within the veterinary track. So I started, I took some time off, and I volunteered at a number of different marine conservation and research agencies around the world. So I worked in Tenerife with cetacean populations, bottlenose dolphins and shortfin pilot whales. I then worked in Central America with whale sharks. So then that kind of renewed my interest in marine as opposed to veterinary. But instead of going to university for marine biology, which is it's not very common back home, I guess there aren't that many universities that offer programs. I did a veterinary science undergraduate degree, which provided me with a scientific basis, like more kind of cell biology, like fundamental principles and infectious diseases, kind of pathology, and that kind of element was my undergraduate. And then after graduating, I knew that I wanted to see about whether or not I could be accepted as an intern. I actually applied for a project student position that Vital got, ironically. So that Hammerhead impact of provisioning project, I applied for it, and I believe PhD Clemency White also applied for it. I got rejected, but the director at the time, Dr. Tristan Gutteridge, sent me a really lovely email and was like, you should apply for our internship program. So I kind of spent all of this time into this application for a project student position and didn't get it. So I applied to be an intern and was accepted, and ironically ended up interning with Clemency, and Vital was there conducting his master's work as that project student, and now we're all working at the Shark Lab kind of 3 years later, which is kind of funny. And I met Ellie at the airport as I was arriving and she was leaving, and
we crossed paths as volunteers very briefly, not realizing we would soon know each other well a year later. And I'm similar, so I'd always had an interest in the marine world. My undergraduate background is actually in animal science, so that's more of a focus on biology and agriculture. But I'd been a competitive swimmer growing up, so I was very comfortable in the water. And then following graduation, I kind of want to dip my toes in as many fields as possible to see where my interest lies. So I did quite a bit of with big cats and wildlife rehabilitation, cetaceans and behavioural studies. And then eventually that led me to the Shark Lab as well. So just like Chessie, I started out as an intern in 2017, start of the year, and then pretty much never left. I've been here on and off ever since. And I think that tends to be a bit of a theme with Bimini. Once you come here, it's very hard to leave. There's a lot to that's attractive about the island and the Shark Lab. So
Yeah.
Yeah.
Do you guys have any other questions? I think there was one question earlier saying, would it be possible to stop the development, or is it for sure going to continue? So I think that was talking about the video I showed you with the development that's happened on South Bimini. It's a very difficult one for us based on the based on the lab. We rely on research permits that are given to us by the Bahamian government, but also the Ministry of Tourism is relying on the money or the income, potential income from those developments for the island. So it's a very difficult position we're in, especially right now in this year with COVID. There is a lot of unemployment in the Bahamas. The Bahamas, I think 60% of its GDP comes from tourism, and with COVID, meaning that the island's been shut down since March pretty much, locals have lost jobs, and there's been an incredible loss of income over this year. So stuff like development and recent development that's happening is obviously a kind of fail safe. There's always going to be people who are wanting to develop. It's a easy job for people. What we can do as a lab and what we ensure we do is we constantly monitor and we put out research. Every year when we apply for our research permits, we have to submit all of the work that's going on at the lab to that government. Saying, this is what's happening, this is the effect we're seeing. So it's very important that we continue to monitor and put out research papers in high impact journals, articles, spreading the word. But like I said, it's a very difficult position to be in, but we try everything we can to ensure that, you know, the development is I guess hard to say, that it doesn't go forward. But unfortunately money talks, and we are a very small lab based on an island where we've suddenly got a new cruise terminal coming in, Virgin Voyages, which is Virgin Atlantic. Rick Branson has just made a new kind of eco tour cruise ship that's all supposed to start and set sail in March. But they've done a load of development for that, so it's really difficult when you're fighting against kind of larger corporations.
Yeah, I think it's difficult, and kind of all research centres and charities are finding this in the Bahamas and also in the Caribbean and neighbouring islands as well. Is the current status of the government doesn't have a particular focus on activism and environmentalism. So we have to find the balance between putting forward the data for these government agencies to interpret, but also taking a step back so that we don't become outsiders from the community. Because often there's a question of what's environmental, but what's also best for people on a local level that live here. So we do have to find the balance a lot, and it's not as easy as just, you know, putting out a big sign saying this is wrong because, you know, so we do have to be quite careful with our wording, and we don't stay neutral, but we have to be respectful of everybody involved in the decision-making process.
And the best way we can do that is support locally run NGOs and agencies that are Bahamian, from Bahamian origin, and words like Save the Bays, which is a big organization around the Bahamas, and Brief, and the Bahamas National Trust, and it kind of put our research out to those organizations, support them, spread awareness about what their conservation messages are as well. But it's a very difficult topic, and a lot of kind of the work we've got here does have and kind of a policy impact. Like Clemency wants to ensure that whatever comes from her PhD data work can be used for informing policy management and stuff. So it's important that always science has kind of a conservation aim and policy involvement as well with it.
I got one more question, and then maybe we should call it good since we're running high on time here. But so this might be better suited for you, Chelsea. What sort of like outreach and stuff do you guys do within the Bahamas? Like, do you guys have like a lot of like educational programs with like schools there, or like children there, or what?
Yeah, so we're really lucky. We have a really great relationship with the schools on the island. So there are 3 schools on the island, and we do open days for them. We provide them with opportunities. So we've had local this year. One of the big things that we've pushed for is to invite local interns. So any of the high school students that are interested in shark science or just want to kind of get a taste of what we do, we pretty much offer them whenever they want to come down, and they can intern. So it kind of crosses over with Ellie's role in managing them. So we have 4 at the moment, and it's really like encouraging them and supporting them as well to kind of pursue careers, whether that's through kind of tutoring them or supporting their work or helping them get jobs, but really provide them with like the opportunity to kind of go out and tag a shark and get involved. And it's really great as well to have like them involved with what we're doing at the station. We also work with Save the Bays and kind of Water Keepers, which are 2 of those Bahamian run NGOs that have programs for kind of the kids on the island, or they'll bring kids to the island to conduct like swim camps or mangrove planting. So basically getting kids out in the environment and showing them kind of why the people pay so much money to come to the Bahamas. It is the habitats that are available around here. And then we kind of do online presentations to schools, and that's one of the big things. We do Google Hangouts with Sharks for Kids, which is run by Gillian Morris Brake, who is an ex-Shark Lab alumni, but she's also based on island on and off. So we run kind of Google Hangouts where we Skype into sometimes up to 1,000 kids in one session in one morning. And we kind of give them a presentation, talk about sharks, talk about the importance of sharks, conservation. And then we offer tours to the locals and anyone who comes to the island. You can come on a Shark Lab tour, and it's normally about an hour, maybe 2 hours if I'm talking, because I talk forever. And we have normally sharks in a semi-captive pen on Back Beach, and we talk about anatomy, and we basically are there to answer their questions, because a lot of people come to the Bahamas and are pretty scared of sharks. So it's really showcasing the beautiful habitat of the Bahamas, the species that are found around here, the work we're doing, and why people should care about them. But basically with outreach, like there's always so much you can do, whether it's online through social media, through presentations into classrooms, through getting involved in local communities like community barbecues, and making sure we have a good relationship with the local people is really important. We have an incredible community here in Bimini, and they're very, very supportive of us as well. So it's also about having relationships with, you know, the fisheries officers on the island and the police and the local fishermen who are bone fishermen. So that's also really, really important for us standing within the community, because we want them to know that, you know, we're trying to protect their environment and the food and the fish that feed their families, basically.
Anyone
I just
have
had a
any
quick.
other-
I do just one quick one. I know with like land animals, when you're trying to tag them in the wild, you get capture myopathy a lot. Is that something that you see in sharks?
What was that? Sorry?
Capture myopathy, where like basically they get so stressed just from the process of being like caught that they end up passing away.
So stress is a really difficult one to measure in sharks. I know it's been done through looking at certain hormones in fish, but with sharks it's pretty difficult. So like cortisol is used to assess like stress in fish. It's pretty difficult to do it in animals, so you do have to look at hormone indicators in blood work. In regards to when you're talking about sharks, Peter, you might be able to answer a bit more about the stress in the animals, but in regards to how we work up our animals at the lab, we actually have very, very strict time limits for the amount of time we're allowed to be working on an animal. So it's 15 minutes really is maximum time you can really have hands on an animal. So that's really from the time limit we want to ensure that that animal swims off nice and strong. We always monitor like how that animal swims off. So it's very short time that we're kind of involved with animal. But when you think about sharks, they are incredibly resilient. That's one thing you would it depends on the shark species. Tiger sharks are definitely more, and lemons are definitely more hardy than great hammerheads. They stress out very, very easily. There's a lot of work going on at the moment about post-release mortality and how likely great hammerheads are to stress out. That's purely if you think about some of how they breathe. They're ram ventilators. They need to keep swimming to be able to breathe. So as soon as you kind of restrain them with a hook, you're keeping them stationary as opposed and giving them very little run, whereas they're an animal that constantly needs to be swimming to be able to push water through their gills. Whereas if you think about tiger shark, they're able to buccal pump, so they can stay still and they can physically push water through their gills. So they do a lot better on hooks. So it really depends on the shark you're talking about. But for us, we are very strict on that time limits, and we know to look for certain signs of stress in an animal. So you look for like pinkishness around the gills. That's really the key sign when you know an animal is very stressed. And we always monitor how that animal is doing. And if anyone on the team or the staff thinks that animal isn't doing very well, it doesn't matter what data we were going to try and get from the animal, we will release it immediately. We never need that data. That's one thing. Did you want to add anything, Peter, about stress?
I thought you might say stress is a very relative thing.
Yeah.
In fish and sharks, just like in people, every species and situation, animals feel it differently, actually. It's how they perceive it. And blood hormones are slightly different in cartilaginous fish than bony fish. And they're usually a correlate rather than an indicator of what's really going on. I've forgotten the original question, but people that respect animals and take care of them properly, as you guys do, it's rarely an issue if you understand them, watch them really carefully.
And yeah, there are key signs that you can look for, and for us, the health and the well-being of the animal is always priority. So if you see any of those signs, we just release it immediately.
Yeah, and ultimately what tells you if an animal's truly stressed is whether it suffers from disease later, which you would see and you don't see, because immune systems get suppressed. That's almost universally the case, and in humans too. People always get sick when they take exams. It's too stressful. It's the case. So I know you guys, you handle your sharks really, really with great respect and care. And Doc always felt that way, and I think it's great. Yeah.
And I think one thing regarding tags, I know you asked us about tags specifically. Surgery or any kind of invasive stuff that happens on sharks have incredible healing capabilities. We still really don't understand the extent of them. There was a paper that came out, I think it was from Jupiter, Florida, a couple of years ago. It was following an adult lemon shark at that dive site in Jupiter. They noticed a bulge in its side, and it's a pretty frequent individual that's found at that dive site, and they were able to video the kind of the lump on the side of this animal over a period of about 1.5 years, and they pretty much documented this shark expelling a metal spearfishing hook through its it's incredible, through its stomach, through its body wall, and the shark was absolutely fine, swimming around like anything. So obviously you don't know what long term, what happens in 10 years or so. But I don't know about you. If I get a splinter, I'm crying like a baby. I don't think I could expel a metal spearfishing hook through my stomach wall, but it just shows that, when you're talking about different animals, you have to talk about that species, like Peter said. So sharks are a lot hardier than I. And if you think about the ocean is a pretty tough environment for them to live in as well. Anyone have any other questions? We could talk about sharks for hours, so.
If no one has any other questions, I'll just say this real quick. I highly recommend going on Peter's trip. I know there's 4 of us officers that did it. We all loved it. I know a lot of people that went on the trip, they all want to go back. Like what they were saying, once you go to Bimini, you never really want to leave. But yeah, so if you ever have any questions about the study abroad trip or anything, you can email me or Peter. You can email the club. We can get you in contact with any of the officers that went. If you have any questions specifically for the Shark Lab, we're oh, I should have asked that, you think about it in half hour or something, email the club and we can relay it to Chessie and she can answer as best as they can. So.
Yeah, my email is pretty easy. It's outreach@biminisharklab.com and Ellie's is admin@biminisharklab.com. So if you have any questions, feel free to email us directly. We're more than happy to
And
answer.
I am too.
And you should come on the University of Minnesota course. It is a good one.
Well, thank you guys for presenting, and we're all very excited and very happy that you were able to. So.
Not a problem. Thank you for inviting us.
Hang in there.
Yeah, stay safe, everyone.
Yeah, stay safe.
You too.
Bye-bye. Thank you, everyone.
Thank you.